Hi, the next speaker is Professor Ronald Lee from Hong Kong U.
So good morning, the UGC committee, RGC panel, commissioner, distinguished guests, all our colleagues and trainees. I would like to, while I'm setting up my computer, I would like to first thank the RGC for organizing the symposium and also for selecting us for an award in the first place. And even though I am the one who is going to be presenting our work, I need to emphasize and disclose that this is really a teamwork consisting of international as well as local investigators. So a lot of thanks to my colleagues and also trainees who believe in the project. So as our project title, Cell-Based Heart Regeneration Implicates, our overall goal is really rather conceptually straightforward, which is really to turn our raw materials into Lego-like heart cells, then mold them together as more complex functional heart tissues or organs that more realistically reflect our native heart. But as sort of our trainees and colleagues will tell you, these are in fact more technically challenging than what you might be going to perceive as I take a more simplistic approach to go over our progresses. So for that, for the panel and also my colleagues who are familiar somewhat with the science, so please bear with me because I'm going to really generalize things quite a bit. But nonetheless, hopefully, my presentation will also offer at least some potential questions that Professor Twain was asking about single cell versus 2D and three-dimensional models. I'm also going to take advantage of the introduction given by HF and so go straight into mine to talk about tissue engineering basically and how we are going to do cell-based regeneration. So the heart is really like a plastic bag. There are 4 chambers with the left ventricular one being the most important one, primarily responsible for blood pumping throughout the body. So heart failure is really primarily basically a left ventricular disease. So now imagine, we have this heart, and now you drill a hole in the left ventricular wall and cut out a chunk of the left ventricular wall tissue. Take a closer look, cross-sectionally. You can see that it's really made up of a bundle of bundles of fibers, and these fibers, in turn, made up of single heart cells, ventricular muscle cells, very nicely aligned in a very organized fashion, just like primary school kids waiting to get into classrooms. But now if you dissociate these fibers into single cells, you can see that they are elongated brick-like cardiomyocyte ventricular cardiac muscle cells, and like bricks, very elongated. And it perhaps makes the most intuitive sense. Let's say if you want to build a very strong wall, you would intercalate rectangular bricks instead of circular bricks together, and that's the idea, and that's also the beauty of the nature. So when one gets a heart attack, there's a block in one of the blood vessels, so there's a lack of oxygen, lack of blood, lack of oxygen, killing off these cardiomyocyte heart cells as the fundamental unit. And therefore, in traditional medicine, as we all know, they do not get replaced, they do not get regenerated. So our project is really about remaking the heart. And we have all the considerations that people who live in the real world have. So number one, what are our starting raw materials for making human heart cells? And how do we mass produce, cardiomyocytes? And when we mass produce, how do we ensure, perform quality control, ensuring that every single one of the cells that we make is of high quality and is safe? When you have a wall, you have a brick, you have a defect in one of the brick, it may not, fall apart right away. But if you get one bad cell, it might turn into cancer, and this is all it takes. And also, when you want to make a car, you have a design. When you want to construct a building, you have a blueprint. So what is ours for engineering a heart? So in our case, we have chosen our raw materials to be pluripotent stem cells. So what are stem cells? And there are a billion different types of stem cell. Really is a collective term. And without specifying which particular stem cell type that we are working with professionally, it really doesn't mean anything. So broadly speaking, all billion different types of stem cell can be classified into adult stem cells or pluripotent stem cells. Adult stem cells are the stem cell types that you can see or isolate from the body of an individual. You know, if they're isolated from the brain, they're called neural stem cells. Isolated from the heart, they're called heart stem cells. And pluripotent stem cells are what we're interested in for this particular project, and they are derivative. So basically, there are two subcategories. One is the so-called embryonic stem cell, first isolated by Sir Martin Evan, who was awarded a Nobel Prize in 2007. Basically, that revolutionized human disease model by allowing the generation of transgenic mice. And induced pluripotent stem cell, the ability to reprogram somatic cells such as urine cells that you have heard about from HF, into the pluripotent state, and discovery was also awarded a Nobel Prize in Medicine and Physiology 2012, jointly to Sir John Gordon and Shania Yamanaka. So what exactly are pluripotent cells, and why do we want to go for this? So it's really like, just a quick analogy, it's really like having a member in your family who can become a doctor when you need one, or a lawyer, or an accountant, or even a chef. And when you have a party, the beauty is that you can have more than one chef, so multiple chefs, and that's the beauty of pluripotent stem cells. So it's really versatile and universal. But the pluripotency itself also presents a major technical hurdle. So, for instance, when you want just a chef, for a dish of risotto, you don't want to have to pay for the doctor fees and attorney fees. So it means that it's simply inefficient and costly. And in heart engineering, about 15 years ago, the yield of generating human heart cells, the efficiency was only about 0.1 to 0.3% when we started, as one of the 4 groups in the world, to be differentiating or making human heart cells. So the next slide are going to be more technical than the other ones that I'm going to be showing. But basically, now I am trying to quickly recap scientifically what motivated us to come up with the present TRS project in the first place. And these were some of the major scientific gaps that we have identified over the year. So for instance, we have found that human ESL, iPSL derived ventricular cardiomyocytes, they are very functionally immature in terms of their calcium handling and electrical properties. And they are also about 10 times smaller than adult human heart cells. And they lack the order organization seen at the single and multicellular levels of the native heart. And they are also, even with the rapid differentiation protocol, they are highly heterogeneous, containing mixed populations of different heart cells. That's because sublineage specifications is poorly understood. And they also do not have, there are also no convenient cardiac chamber-specific surface markers for robust purification, unlike neuronal cell and blood cells that we all are familiar with. And when you transplant them, the graft survival is pretty poor, and the immunobiology is also very poor. And given all these uncertainties, the field isn't quite sure about the safety and efficacy. So as such, we came up with 4 projects to address these questions. And basically, this is a basic slide that I'm going to walk you through. So with somatic cells isolated from patients of different heart conditions, we can reprogram them into iPSL. And HF has been using urine, and we've been using blood. So meaning that we can get some blood sample from you and reprogram them into these pluripotent iPSL. And together with human embryonic stem cell, we can specify or mass produce ventricular, human ventricular heart cells as single entities or fundamental units for carrying out some basic studies to look at their metabolism, electrical as well as mechanical properties. So it's very important to understand the basic biology because this is the only way that we can do cell engineering and therefore improve the properties of these single cells. So with better cells, we can make better tissues and better heart organs chamber that I'm going to show you. So these single cells can be put together as two-dimensional construct that we call cardiac patch or disc, or even three-dimensionally as muscle fibers or chambers. With such, we can use these tools for modeling diseases such as Duchenne muscular dystrophy, and also for drug discovery and cardiotoxicity screening, meaning that we're using these as in vitro diagnostic tools. And as we go further, we would like to, and the very same constructs can also serve as prototypes for future transplantation. So we also have a project for looking at the preclinical potentials as well. So the progresses that I'm going to be presenting to you over the next 10 minutes or so are going to be primarily focusing on project one, which has to do with engineering heart constructs. And we are taking the critiques and input by the panel very seriously about restructuring our project so that we can be hopefully achieving more strategic outcomes in the near future or with the remaining support for funding period. So this is, we're looking at a colony of human embryonic stem cells. So there are, this particular colony consists of about 10 to 50,000 cells. And this slide, basically, I want to show you that we can mass produce human ESL, iPSL-derived ventricular cardiomyocytes. So as I said earlier, about 15 years ago, I apologize, it doesn't show up so well, the color contrast. So about 15 years ago, as we got into the area, the yield was only about 0.1 to 0.5%. And now, if you go into the literature and the latest conventional technology, you can generate about 1 to 2 cardiomyocytes from each starting undifferentiated cell. And with the TRS support, we have developed a new protocol allowing us to generate about 60 to 70 cardiomyocytes per starting undifferentiated cell, representing about 2 orders of magnitude or improvement over the old protocol. And it's extremely important because this fundamental need, the ability to mass produce ventricular cells, allow us to do tissue engineering. We don't have cells, we don't have the fundamental units, we cannot engineer anything at all. So with this video, you can see that about 100% of these cardiac cells, if you will, are already beating, and each consisting of about 10 to 50,000 ventricular cardiomyocytes. But we are not happy with them at all. And in fact, we think they are functionally misleading because they are very random structures. If you look at the histology here, there's nothing like what you see in the native heart. So in many ways, it's like you go to Toys R Us and buy a heap of Legos, and before you open the bag, before you get them out and put them, put the single pieces together, they're really nothing but raw materials, and this is the same idea here. So it's just a heap of cells. We have to somehow disintegrate them and then put them back together, and this is exactly what we did. And here we just enzymatically digest these balls of cells into single cells. So, and you can see that these single human heart cells are very immature and they are rounded. So it's not quite like the brick-like elongated adult cells that I showed you earlier. So we are definitely not happy with them. And with a series of studies looking at by using a combination of biological and physical approaches, by looking at the epigenetics, physical, fusion, neurohormonal inputs, and also a bunch of other micro-environmental non-cell autonomous factors. These studies have been published during the duration of the past 2 years of support. And we are able to stretch and drive maturation or hypertrophy, making the cells bigger and longer. So mimicking what we see in the adult heart better. Not the perfect solution yet, but we are getting there. So at least they look better as single cells. And then we can put these single cells, rearrange them using nanotechnology or microgroove technology, rearrange these single cells, put them together as aligned two-dimensional cardiac patch or disc. These constructs, you can see that alignment is there, and a lot better than the random structure that you see, with regular in vitro differentiation. And why is this important? As it turns out, by making these constructs, we are also learning more about the basic biology. For instance, we know that by aligning together, we can generate bigger forces. And actually, it makes a lot of sense, because if you play a tug of war, and you want the player to be lining up together, pulling in the same direction, generating the forces instead of going in 50 other different directions. And also, we have also noticed quite significantly as electrophysiologists, this is our primary interest, the constructs, when the cells align together, we are also able to reduce the incidence of arrhythmias, and these are the kind of functional assays that we can look at. So we're not looking at single cells anymore. We're looking at two-dimensional patches here. And from 2D, we go 3D, and here we're looking at some hair-like human heart microtissues. And the video shows you that about 500 human ventricular cells, they come together spontaneously to form these so-called human heart microtissues. And this is top view, this is the side view, and this is really about the size of the human hair. And the beauty is that, look at this, we can put about 100 hair in each of these single wells as an array for potentially high throughput screening, looking at different conditions, looking at different drugs, etc. And if you go to a cardiologist for a stress test, HF would put you on a treadmill and run. And how do we do that at the single cell level? And here is what we do. And by putting a magnetic bead on one end of the muscle, we can put this under a strong magnetic field. Then we can kind of like stretch it mechanically to see how durable and how they respond biologically to mechanical stress. So these are hair-like structures, very fine, very small, consisting of about 500 cells. And you can argue that this is not physiological enough, and therefore, by working with Kevin Koster from Mount Sinai in New York, we are able to generate a trabecular-like muscle strip like this. This particular one is about 1 centimeter in length with about ½ centimeter diameter. So literally, it's very hard to tell even if you had to isolate a native trabecular muscle from the native heart. So you pour cells into collagen matrix, and this is how you make it. This is the side view. And the most interesting or most exciting of all, perhaps, is the mini human ventricle that we have managed to make. And you can see that this is contracting. And this is about the size of your thumb. And the beauty is that, three-dimensional, and for the first time, if we put a catheter in there, we're able to measure hemodynamic, pressure, volume, loop, etc. So, and also electrophysiologically, we can look at arrhythmias, etc. So perhaps this is one of the areas where we can cross with other ongoing efforts, such as the one that HF just presented about personalized medicine. So to summarize this part of the talk for Project 1, meaning that I can take urine sample, blood sample from you, and you give us like 4 to 6 months, you can come back and get your personalized mini heart. And the idea is that we can proceed to kind of like look at personalized medicine, because some monogenic or even drug-induced diseases, their drug responses, for instance, depend on the polymorphisms. And the polymorphism depends on whether you're Caucasian, Indian, Jewish, Chinese, different ethnic groups, etc. And in the very near future, if it's not impossible, it's going to be very hard for the scientific community to figure out what these polymorphisms do. So in this case, you can use a personalized, for instance, in this case, personalized mini heart to screen drugs and see what side effects are there, whether they are pro-arrhythmic or not, and find out sooner before they are being used in patients. So for instance, with these models, we're able to pick up flecainide as a pro-arrhythmic drug, which flecainide is the one that has been withdrawn from the cardiac arrhythmia suppression trial. So as we go forward, we want to be able to further fine-tune this technology platform and improve the sensitivity, improve the accuracy, so that we can use these tools to assist drug discovery and also disease modeling. So I'm going to make a rather long story really short, and basically, sorry, let's skip all the animation. So with Project 2, we've been focusing on looking at the cell biology, and basically for adult cell, you can see that there are the so-called T-tubules, which really are invaginations in the membrane, serving to bring critical ion channel proteins close into proximity. And this is completely absent in human ESL-derived ventricular cardiomyocyte. And also there are a bunch of critical calcium handling proteins that are robustly present in adult cells, such as triatin, junctin, SERCA, and calciquestion. They are completely absent or very lowly expressed in a human ESL-derived ventricular cardiomyocyte. And then there are other proteins that are not so highly expressed in the adult cell, but they are more highly expressed in the human ESL-derived ventricular cardiomyocyte. And as I said before, it's very critical to understand the biology of these cells so that we can go on and engineer and improve the properties of these cells as fundamental units for making better tissues. This really is my favorite slide, summarizing the basic knowledge that we have learned from these studies, but for the purpose of this talk, I think it's probably too technical and busy, but for those of you who may be interested, I'll be happy to go over with you after the talk. And Project 3 is about enhancing the therapeutic potential. With this, we've been looking at the immunobiological properties of the derived cardiomyocytes, and also this is an ongoing effort that Professor Campbell-Healy is undertaking by looking at the cell surface, using the cell surface capture technology. He is trying to probe using a combined omic approach by looking at the epigenetic, transcriptomic, proteomic, and a cell surface proteome. Using an integrated combined omic approach, he want to be able to barcode different progenitor cell populations. This is a way to ensure quality control. And preclinical translation, actually, this is, we have built models, animal models for the so-called six-signer syndrome model, and also heart failure model in pigs. And these are available for us to move on and to transplant. And perhaps one of the technologies that have been more mature is the so-called artificial pacemaker. And we've done quite a bit of experiment using gene-based approach, and now we are looking at a progenitor population, hoping that we can isolate pacemaker cells for transplantation. And these are ongoing, but like I said, we are taking the different directions, and we need to invest in those that may be more fruitful for the remaining period of the grant. So together, we are a new group. We did not exist about 3 years ago, and with the kind support of the TRS, we have recruited and we are training about 40 staff, postdocs, pre and postdoctoral trainees. And so far, there are about 40-something publications with about 12 as invited reviews, and 5 others were enabling technologies that were developed before the grant started. And we have a few other ones submitted. And so as we go forward, we really want to be focusing on improving the quality of our work instead of just looking at the quantity. As infrastructurally, with the TRS support, we have built multiple cores. And at the HKU, we have the culturing and differentiation core, reprogramming core, gene vector core, and a functional core with robotic setups to look at electrophysiology and also calcium imaging. Humanized mouse model, and omic bioinformatics core. There are 2 components. One is the HKU, the other one is the CUHK. And at HKUSD, they focus on ultra-high resolution bioimaging, and also there's a bioengineering core for generating some of the constructs that we are using for tissue engineering. CTU is the biorobotic, has the biorobotic core, and CUHK the mechanobiology core, and also, as I said, the other component of the omic core. So the 4 universities, the 4 components at the 4 universities work together as the Hong Kong group, and with such, we work collaboratively with several US institutions, including Harvard, Mount Sinai, NIH, Stanford, and lately we've been developing very significant effort with the Hopkins group as well. So this is basically what I wanted to say for this presentation, and I'll be happy to take any questions that you might have. Thank you.
Thank you very much for the exciting talk. Question for Professor Lee?
Thank you for a very nice talk. So what is the strategy to bring this, technology ultimately to benefit the patient? Are you thinking of sort of cell-based therapies, or are you making artificial organs and then through surgery,
We have in mind a timetable, and even just for Project 1 alone, we have filed altogether, or in the process of filing altogether, 8 to 9 IPs, with half of them involving Hong Kong U. And realistically, we are immediately thinking about taking advantage of these platform of tools to develop better in vitro diagnostic tools for drug screening, improving drug discovery. And like I say, the possibility is really unlimited if you consider the ability to reprogram. For instance, an advantage that we have here in this dual political space is that we have access to different Chinese ethnic groups. And I talk about polymorphisms and the importance of that for, figuring out the side effects of different drugs or ongoing drugs, that our cardiologists routinely prescribe to different patients. So, and combining the different ethnicity with disease conditions, I think the possibility is really unlimited, and that will allow us to immediately take advantage of these tools for personalized medicine, not in a sense that you put these constructs immediately into patients, but to allow you to look at drug effects and side effects, and whether by using these tools you can discover new drugs more effectively. And the reason why I'm not saying about cell-based therapies yet is for obvious reasons. There are regulatory concerns, and also I don't feel that the field is quite mature enough for us to think about that yet. I'm not saying that the time won't come, but just not right now. And especially for repairing the myocardium, you're talking about a lot of cells in a highly organized manner, and even the cell delivery method, there isn't really a good consensus yet. But the time will come, probably several years later, as we learn more. However, if I am to tell you one possible scenario that we may pursue clinically, that would be the pacemaker, because all we have to worry about is really putting about 3,000 cells into the atrium, and we can have possibly the first the prototype is going to be a hybrid system. Pair it together with an electrical device, and just in case the artificial device fails, the electronic one is going to take over. But that's because of the reason that we are talking about very, very small number of cells, only 3,000, because the native acid node, it consists of only about 3,000 cells. So we have a timetable for thinking about this.
So I think that one of the interesting, it was a very good talk, by the way, thank you very much. One of the interesting things that you brought up was using these cells for drug screening, because you've rightly identified that there may be ethnic differences or ways in which different groupings may metabolize enzymes. To some extent, that may be also related to liver function. And it would seem to me that there's a very good synergy, potential synergy between the two groupings. And I think that's a very exciting prospect that there is this possibility of increased interactions in a broader scale, bringing different themes together.
We would like to cross-talk more among the two projects. Absolutely. Yeah.
Thank you very much, Ron, for a very dynamic talk. So let me go back to the hard tissue engineering and regeneration aspect. So as you know, this is a extremely active field, and there are people literally all over the world in various institutions and research and universities that are hard at work, and they all have their own gospel to preach. So number one, so how are you? I guess where do you see your one-upsmanship on some of the other groups? Number one. Number two is that, as you also well aware, the use of adult stem cells for myocardial infarct. There are also people who are
Absolutely.
earnestly
Mhm.
convinced that that is the way to go and forget about all these pluripotent stem cells, which have other issues which you already raised. So what would you say to them if they're sharing the stage with you? Again, there's no sure path to salvation at this point. So everybody can preach their own gospel. I just am curious what you would say.
I understand. Before I answer the question, there's a confession that I need to make is that I personally dislike the idea of claiming that we are the first of doing such and that we are the most competitive group in such areas. I think this is for the scientific community to judge, not for us to say. But with that said, I think our niche would be to focus on electrophysiology, and I think that this is an area that we feel most familiar and comfortable with, and this is really our edge, and we're not going to give it up just because there are other, seemingly interesting areas of jargons or, buzzwords that people like. I mean, we might combine the technology, but still, we want to take advantage of that. And the second question about adult stem cell. So I'm fully aware of the data out there. And I would like to say that I take a more open approach, meaning that I think the idea is great, but I'm not quite sure that the data out there are convincing yet. What I'm saying is that, I think both are good options, and it's good for us to keep an open mind, a minded approach. But we have chosen to study human ER cell, pluripotent cell, because we have only very limited resources. We don't want to be spreading ourselves too thin, looking at this, looking at that. But while we are looking at that, I'm not excluding the possibility that the other is also a good option. But in reality, I mean, later on, I think it's good for patients to have options as well. We have Panadol and we have Tylenol. It's good to have some options. But scientifically, but like I say, I can be very blunt, about I'm not sure if some of the ongoing experiments are what we want to see or they are convincing enough. But it has nothing to do with the area, just how people are interpreting or conducting the experiment.
Yeah, I think it's a big world, so there are many roads to the same target. So then I forgot to mention one more thing, and that is there's another group of people out there who are the tissue-specific matrix, extracellular matrix group, who some of the religious leaders in that field would tell you that unless you have tissue-specific extracellular matrix, you can forget about guiding a kindergarten baby that you have shown in swaddling clothes to ever develop into anything that you desire. Again, that's an extremist view, but some of the fervent leaders will tell you that.
Sure.
So given that, I mean, they also have published papers and whatnot. So I'm wondering whether you do. I understand you have limited resources, but still, what's your view on that? Just curious.
Sure. I mean, everyone is entitled to have their own opinion, and I think the jury is out there. But our approach has always been a reductionist approach. So we start with something simple, and we are able to show that we get these contracting constructs, tissue strips, chambers that function and work, and we can make reasonable physiological or close to physiological parameters. But does it mean there's no room for improvement? I think there's a lot of room for improvement, and we have to confess that we don't have the immediate access to the expertise in biomaterial. So this is an area that we definitely can do better. But how sophisticated? I mean, like I say, we're starting with something simple, something that works, and then from there we're going to build from there and add different levels of complexity and see which are more critical and try to figure out what are more important in the process.
Thanks.
Any more questions? If there's no more questions, let's thank Professor Lee once again.